Nick Hastie will tell us everything you ever wanted to know about WT1.
This has been a wonderful meeting, and the quality of the talks has been outstanding, as far as I'm concerned. And it's great you guys are here, but this deserved a much bigger audience. And perhaps it will go on some website. But this is really high quality stuff. And perhaps there's other things on, perhaps, I don't know what's going on, but still. I feel I'm an imposter at this meeting. A lot of it was nice that we didn't have everything on hematopoiesis and leukemias and things, but I am going to talk about, I suppose, a major transcription factor, whatever you call it. What did you call them? Master transcript. But my story goes, and something will be about cancer directly, but quite a lot will be indirect in relation to cancer. And what I will show is how many things a single gene can do. So at the end you'll say, my God, that's ridiculous, probably. People say that to me. But really we're revealing more and more things about what a single gene can do. And it's not just transcriptional, it's downstream, binding RNA and doing a whole lot of other things. So that, you know, there'll be a lot of biology, it'll be too complicated, but I'll try my best on this, okay? So, the starting point, I'm in a human genetics unit, is human genetic disorders. And is there a pointer? Oh, is this it? So, many years ago now, when I moved into a human genetics unit, I got excited by this problem because I wanted mainly to get into developmental biology in relation to disease. How do I press it? What do I press? Yeah. There's lots of little things. That one?
Probably the other way around.
The other way around. Oh yeah, it's good to press it. I put it in somebody's eyes. Yeah. So the starting point for what I've been working on is this childhood cancer called Wilms tumor. It affects about 1 in 10,000 children. It's about the most common pediatric kidney cancer. And these tumours can grow up to about 2 kilograms in size in a child. And the reason they fascinated pathologists for over 100 years is they're probably the best example of cancer, the first best example of cancer arising through development going wrong. And what we think happens is there's a single mesenchymal progenitor cell that normally should differentiate into the epithelial cells of the nephron. And because it has a genetic problem, it could be loss of WT1 or other problems, these cells proliferate, they can't differentiate properly. In some cases, they try to make kidney structures. They make bits of glomeruli, they make bits of epithelial structures. They try to make a kidney. In other cases, they can't. And in the cases I'm talking about, 15 to 20% are caused by loss of function. This is a tumor suppressor, not an oncogene. By loss of function of WT1. You can have children who inherit a mutation in one copy, they lose the function of the other copy in a mesenchymal progenitor cell, and those cells will expand to form the tumor. Now what's different about this subset, they are mainly mesenchymal. They don't form epithelial structures. And often they can have muscle in them, they can have bone, cartilage, and fat occasionally. These are very unusual tumours. And you'll see why they get these structures later if I remember to say. So, early on, we thought this is a problem in kidney mesenchyme not being able to differentiate, and I'll come back to that. So how can I justify? It's interesting to hear quite a few people have been a bit gene-centered in this. It's supposed to be bad to be gene-centered, it's supposed to be process-centered. But in this case, this gene's led me into a lot of biology, I believe. So why have I been interested in so many years? Well, first of all, the clinical side. Loss of function mutations, as I said, can lead to Wilms tumours. These nearly always have gain-of-function mutations in beta-catenin, these same tumours, and sometimes IGF-2 overexpression as well. Now, there's several syndromes in which inherited mutations in this gene can lead not only to Wilms tumour, but severe glomerulosclerosis, which can lead to death in the child, gonadal dysgenesis, including sex reversal, and sometimes heart disease and congenital diaphragmatic hernia. And WT1 is not expressed in many adult tissues, as I'll say, but it is highly expressed in a number of adult cancers. It's hypothesized to be an oncogene. There's not, in those contexts, there's not very good evidence. And it's mutated in some leukemias, including AML, for example, we've heard a lot about today. So we'd like to know the mechanisms underlying these, of course. But the second thing I really wanted to work on this for was developmental. If you knock this gene out in mice, you get no kidneys, you get no testes, no ovary. And as we've shown, the heart can't form vasculature, and I'll explain why, and the mice die because they don't have a blood system, basically, in the heart. Now, in the kidney, it does lots of things, this gene, at different stages. But the thing we've concentrated on, I will show, it's required for the mesenchyme to epithelial transition, essential for making nephrons. But in the heart, it does the reverse. The heart has a layer of cells called the epicardium, a mesothelium around the heart. Budding out from those cells through an EMT, or mesenchymal cells, they are the progenitors for the coronary vasculature. And WT1 is required for that EMT, and I'll talk about that briefly. Now, more recently, though, we've been very surprised that this gene is vital in the context of adult tissue survival and function, and it's induced in a number of tissue repair processes, which I'm going to talk about as well. So my talk will move from kidneys to hearts to fat to liver fibrosis and back to tumours at the end. So I move around quite a bit. I'll try and make it sound logical. No, but the other thing that's really fascinating me for you is this gene encodes multiple isoforms, and two isoforms, just differing by 3 amino acids, have very different biochemical functions, we think, as well. So that's the introduction. Press this. So what sort of proteins encoded by this? Well, and in mammals, there's at least 24 isoforms. I won't go into that because only 2 seem to be very important. It encodes a zinc finger protein with 4 zinc fingers. Each of these zinc fingers binds 3 DNA. There's also transcriptional activation and repression domains. It's a transcription factor. All other vertebrates have a WT1, highly conserved, and there's 2 isoforms that only differ by 3 amino acids, lysine, threonine, serine, inserted by zinc finger 3 and 4. There's more of the form having the 3 amino acids. And Ken McElreevy years ago, in the past, showed in a syndrome called Fraser syndrome, if you have a mutation that affects the splicing of just one allele, so you don't make the KTS from that allele, you change the balance, these children get glomerular sclerosis, and the males develop as females. So the balance between these 2 isoforms is crucial for function, and these are conserved in the right ratio over 400 million years of evolution. So those are very important, and as I'll suggest, they probably have very different functions. So just to get an idea about tumours and various other things, where's this gene expressed? Well, mainly in the kidney, the gonad, the mesothelium, which I'm going to explain, quite a lot of body mesenchyme, and some of the sensory nervous system. These tissues all arise from the intermediate and lateral plate mesoderm. And if we look at a differentiated testis or kidney, or in the adult, you will see expression in the kidney in this beautiful cell type called a podocyte, which has long processes, wrap around blood vessels, and that's where filtration takes place. WT1 is essential for the formation and maintenance of this structure. This is the Sertoli cells of the testis. It's essential for those as well. But this is a thing nobody's ever heard about, apart from mesothelioma. This is a mesothelium. It's a stunning thing, as I'll try to describe it. Your body lining and your visceral organs all have a mesothelium. And for years people thought this, and the heart is called the epicardium. And for years people thought this was just a protective layer that helps ionization, protects against infections of tissues. But actually, as we and others have shown the last few years, this is a source of mesenchymal progenitors through an EMT, the cells migrating to the tissue. And as I'll show, in the fat, they form visceral fat progenitors. In the liver, they form stellate fibrosis cell progenitors. And in the heart, they form vascular progenitors. And I'll describe that a bit. So these are beautiful, wonderful cells. Probably most people don't even know about. Now, what's special about these cells here? They're all called epithelial. But very unusually, they're epithelial with mesenchymal properties, and that's why they're called mesothelial. So they're somewhat plastic on the boundary between 2 cell states. And WT1, expressed in all these, WT1 is very important in maintaining the mesenchymal or epithelial nature of these cells. Right, so a bit about kidney development. Kidneys develop from 2 embryological sources, both from the intermediate mesoderm, the ureteric bud, which is going to form the branching collecting system, the mesenchyme condenses around the bud, here, And this mesenchyme is going to go through an MET to make this beautiful nephron. You have about 1 million of these. And a vital step in this is Wnt4, the Wnt signaling molecules, Andy McMahon's lab showed, is sufficient and necessary to induce this MET to make the nephrons. We've hypothesized that it's this process that is effective in making the Wilms tumors with W2 mutation. Where's W2 expressed? We saw years ago, it's only in the nephron, low levels here. The levels induce dramatically before Wnt4 comes up, and they stay on eventually just in those podocyte cells I talked about. So, if you delete, as Jordan Kriegberg did, if you delete W2 in total null, you don't get kidneys because this very first step can't happen. These cells don't survive in a complete deletion. And what Andreas Schedel's lab showed beautifully is W2 regulates FGFs and BMP signaling to allow these cells to survive. Now, we also know W2 is essential through mouse and human genetics for the formation and maintenance of this podocyte structure. And recently, Andreas Schedel, Jordan Kriegberg, and Christoph Engler has shown W2 transcriptionally activates a whole series of podocyte structural proteins, which themselves are mutated in human disease. So it's a master regulator of these two steps. But we wanted to know what happens here. We think tumours arise here. So we had to make a conditional mutant, which we did after a lot of struggle. We made a flops W2 nano. We deleted it in nestin Cree just in here. And what happened was, as we expected, we did not get nephrons. The mesenchyme amplified. The mice died. You can't survive after birth without a kidney. One mouse did survive and he got a tumour. But usually you need more than just W2 mutation to get tumours. So if you delete it in here, you can get a tumour. Now, dramatically, the levels of Wnt4 were reduced completely. And as we've shown, W2, a key thing it does is transcriptionally activate Wnt4 to drive the MET. So now I'm going to describe the yin-yang of, not all published, partly because the postdoc who did this work had a terrible accident. He was paralysed from the waist down. He's got a job as a lecturer. He cannot continue the experiments yet. And this is for several years, for some of these. So some of the best experiments are still not published, I'm very sad to say, but most of this, I suppose, is. The principle is, this is a remarkable situation, I think. I hope you agree. In the kidney, in the cap mesenchyme of the kidney, W2 is required, as I said, for the formation of an MET that drives differentiated neurons. And we've shown it transcriptionally activates both Wnt4 and the cell adhesion molecule E-cadherin. You heard about the E-cadherin. Now, in the heart, what we showed, we deleted W2 specifically in the epicardium. And now, we didn't make vascular progenitors because we didn't get the EMT that goes on to make vascular progenitors. So in that case, we showed that W2 transcriptionally activates the major EMT protein, snail slug. So it activates snail and slug. In the kidney, it activates Wnt4 and E-cadherin. But in the kidney, going through the MET, it represses snail and slug by binding to the same site it does when activating it here. And in the heart, it represses Wnt4 and E-cadherin. So you've got this reciprocal activation repression that makes sure, in one context, you're going to do an MET, and in another context, you're going to do the EMT. If we delete W2, we reverse the process, so these cells that should have become epithelial will amplify to become a mesenchymal Wilms tumor. And it's interesting, we heard about LSD1. One of the unpublished things we don't have, in all contexts, it requires LSD1. As an activator or a repressor, it needs to interact with LSD1. And W2 activates or represses a whole chromatin domain around these structures. It involves an opening or shutting chromatin as well as being a direct transcription. So that's part of my introduction, really. So this thing is important in development, and it does it as a transcription factor to some extent. I wanted to know, does it function in the adult? There's a lot of speculation about some of these processes that are active in the adult. EMT is activated in tumours and tissue fibrosis. Is it required normally for some of these processes? And is W2, when children get glomerular sclerosis of the kidney, is that a developmental problem? Or do you need to keep this gene on in the kidney to maintain its function in the adult? So what we did, actually Klaus Nelov is on this paper, he helped us with some of it. We deleted, my postdoc there, Nguyen Chow, deleted W2 everywhere. We did all the controls, pre-controls and everything else, in an adult mouse. Now the gene's only expressed in the mesothelium, the podocytes, the glomerulus, the cells in the gonad, and 1% of bone marrow cells. So we didn't expect much. We might have expected a kidney phenotype. But what we got is very surprising. The mice died within 10 days. This is severe glomerular sclerosis of the kidney. And what happens is the podocyte foot process has just disappeared. The kidney can't work anymore. That's probably why the mice died. But surprisingly, very quickly, the spleen went down to a third its size. You no longer made red blood cells in the spleen. 80% of the exocrine pancreas died through apoptosis. And we lost a lot of bone and a lot of body fat. This is micro-CT. Body fat, we lost a lot of body fat. So a lot of this is surprising. What's going on here? This is cell autonomous. W1 regulates podocyte functions. This, we think, is local paracrine. We think stellate cells are being activated that express W1 when you delete it, and you activate cytokines that kill. Some of this is systemic. The levels of IGF-1 go down, circulating levels go down 95% in this mice. But the thing we really wanted to get our hands on, is it in these mysterious fat cell progenitors? Because it's a really hot area, and we wanted to know, could this play a role, identify fat stem cells and progenitors? And the answer is yes, we've published this, but so I wouldn't put this politically incorrect type of slide, my postdoc Yo-Ying did. So there's two major types of fat. And there's visceral fat. Two major types of fat, white and brown. There's not much brown fat. That's thermogenic. It's very important. It gets rid of fat. And then you have subcutaneous white fat, a lot of that around your body, and then visceral. And visceral is on your organs, like the kidney, the gonads, the heart. You have these visceral depots of fat. If you have too much visceral fat, it puts you at risk of cancer, heart disease, type 2 diabetes, and possibly dementia. This is probably some of the most relevant work I've done in relation to disease, because what people didn't know, if you have major types of fat, do they come from the same progenitors? Where do they come from, these fat progenitors? And so, Rodehefer in Jeff Friedman's lab published a cell paper where you can actually sort progenitors. I won't go through this. You do a series of fat sorts, and you take this from the SDF, stromal vascular fraction of fat, has progenitor cells. There's very few of them, and you go through all these sorts and you find them. Now, to cut a long story short, WT1 is only in these cells, the pre-adipocytes and the progenitors. And it's only in the ones that are for visceral fat. It is not in the subcutaneous, and it's not in the brown fat. So now we've identified specifically a marker for progenitor cells in visceral and the bad fat, and not the good fat. To prove these are really progenitors, I'll go through this quickly, you do a lineage tracing. Everyone know about lineage tracing here, probably? Does everybody know about, hands up who doesn't know about lineage tracing? Somebody? So what you do, it's no good just sorting cells and saying that they are progenitors. You have to show those cells led to the production of fat in this case. So what you do, we got from another group, WT1 Cre-ERT2. So you activate pre-recombinase where WT1 is expressed with a pulsar tamoxifen. You cross that to a reporter line, which expresses everywhere a tomato color. But if you do a Cre-deletion where WT1 is expressed, you activate GFP. And any GFP cells, and its constitutive promoter, any GFP cells would have come from these WT1 cells expressing. And to show the most dramatic example of that, these cells in a 1.2-year-old mice, they are gonad fat. This is supposed to be GFP. She labeled it red just to be difficult. 80% of this fat in a 1-year-old mouse came from a pulse of tamoxifen activated WT1 at E14.5. So that fat came from development from progenitors. Where do those progenitors come from? Well, what Yo-Ying found was fat cells, visceral fat, has a mesothelium, just like the heart. And we showed that it's this mesothelium that produces the progenitors. As it does the vasculature in the heart, it goes in and makes adipocytes. Oop, I'm tripping over my laces, which have come undone. So This is shown very nicely here. We culture some mesothelium from the gonad, where fat is going to be made. We put in lineage tracing. So the green fat cells came from the WT1 population, the red didn't. So there's heterogeneity. Some cells come from one set of progenitors that came from this mesothelium, the others came from another set, and we don't know where they came from. But this is Raman spectroscopy, and we show the fat coming from WT1 progenitors has different properties than the fat coming from the other ones. So you can have heterogeneity of progenitor cells, and the fat that comes can have different properties from them. Right, so these are some of the conclusions from that. The bad fat and the good fat have different origins. The visceral fat comes from the lateral plate mesoderm. There's heterogeneity. There's different types of progenitors in different fat bodies. There's a mesothelium, which is a source of some of the adipocytes, and this says WT1 has a broader role in mesenchyme regulation than just in the heart or the kidney, whatever. It's doing more, and it's probably doing it in the bone as well, but we haven't looked at that much. So, most of my talk's molecular biology. I haven't got, sorry, to that. I'll rush through that. It's all about RNA. This is just briefly to say, as well as being involved in development and adult tissue homeostasis, this protein's activated. When you damage hearts, it's activated in the epicardium. We just showed a paper in eLife. If you damage the notochord in zebrafish, a population of cells coming from on the sheath that lines the notochord migrates in and helps repair the damage of the notochord so that vertebrae get produced properly. So you can look at that paper. I'm not going to go over it, but it's very important in this notochord repair process. But what I am going to mention, and this could be, again, the most important thing for cancer that we're working on, is WT1's induced during hepatic fibrosis. So, what's hepatic fibrosis? If you drink too much, and we try not to, I try not to drink Monday, Tuesday, Wednesday, Thursday. But my son-in-law came for dinner the other night, and I broke that rule on Tuesday. So in there's a population of cells called quiescent hepatic stellate cell, this beautiful cell with long processes again. What happens when you reduce fibrosis? It can be through too much alcohol or various other insults. These cells change in shape, they produce an extracellular matrix, and they produce a scar. And that scar is supposed to heal the damage. But actually that scar is a double-edged sword because that leads on to cirrhosis of the liver and liver cancer. So this stellate cell is a good and a bad thing, but in this context it could be bad. I'll go through this very quickly. So what Tim Kendall, who's now a consultant pathologist, showed, this is a liver after damage with carbon tetrachloride, which mimics the effects of alcohol. Suddenly you induce WT1 expression in exactly the stellate cells where the scars have been formed. Now, these red cells are also stellate cells, so again there's heterogeneity. Only half the stellate cells express WT1. And what we've shown is these cells are quiescent. They came from the mesotelium again, early on in development. They migrated into the liver, they sit there not expressing WT1, you reduce fibrosis, WT1 is activated, and we've shown that these have a different shape than the other stellate cells, and they express different profiles of gene expression as well. And they come from the mesotelium. Now, when you look at the genes that are activated when WT1 is activated, it's remarkable. What we get is response to wound healing, extracellular matrix remodeling. The levels of change in the expression of the genes are 500-fold for longer. We're talking massive induction of RNA and protein in these cases when you activate these. So what's WT1 doing? We delete WT1, and this might not look much to you, but this is a big effect. Paradoxically, it identifies the most fibrotic cells, but if you delete WT1, fibrosis goes up. And that's quite dramatic. That is a big increase in tissue fibrosis. And the way it does that, I haven't got time to go over it, so it's a fibrosis suppressor as well as a tumor suppressor. And the way it does that, because we've done the transcriptomics, it remodels the extracellular matrix and activates Wnt signaling. So you delete WT1, you get more fibrosis, and you get dramatic remodeling of the extracellular matrix and upregulation of Wnt signaling. So it's a major factor in fibrosis, suppresses fibrosis, and these cells come from the mesotelium again. And just to summarize the mesothelium, I had a little review in development last year. The mesothelium in the heart, the epicardium, produces some vascular cells, some, not all. In the liver, it produces some stellate cells, not all. In the fat, it produces some adipocytes, and not all. So this leads you to knowing there's tissue heterogeneity in all these cases. We don't know the functional reasons for that. And in the intestine, my friend Ramon Munoz-Capelli showed, same again, you make interstitial cells. The pacemakers of the intestine come again from the mesothelium in the intestine. Right, so how does W1 do all these things? It's clearly a transcription factor, but we showed, I'm so old, this is over 20 years ago in a Cell paper, we showed that W1, the different isoforms localized differently in the cell. The plus KTS form associates with splice complexes and the minus with the transcription apparatus. We went on to show W1 integrates with splicing factors, active splicers, sorry, and it interacts with splice factors and it interacts with RNA as well. And the plus KTS form seems to be better at interacting with RNA than the minus. And the other thing we showed is this protein is nuclear, yes, but you've got to be careful, because it shuttles forwards and backwards between the nucleus and cytoplasm. All the isoforms load onto polysomes and then go back into the nucleus. So we hypothesized that perhaps this thing is unusual. It's not just a transcription factor, it might be involved in several stages of post-transcriptional regulation, and that's what we're showing now. Now I'll try to go through it quickly. We stopped working on this because we did not have endogenous RNA targets to which it bound. Now we do, as I'll show. But just to show our recent proteomics, and a lot of these have been validated, this protein interacts with transcription factors, splice factors, and the M6A methylation protein, WTAP. It interacts with transport molecules, RNA turnover molecules, arginort involved in microRNA function, and translation factors. So we hypothesized, perhaps this is very unusual, that it's actually involved in gene expression all the way from transcription through to translation, and perhaps it's chaperoning some RNAs all the way through the process. But we had no way of testing that because we had no RNAs, so now we have used two approaches to identify endogenous RNAs interacting with W1 in two cell lines, an ES cell lines expressing W1 and a mouse kidney cell line, mesonephric cell line expressing W1. We've used two approaches to get RNAs. One is just to immunoprecipitate W1 associated RNAs and sequence them, but the other one is something called FLASH. You cross-link your protein to RNA and you sequence. This not only maps the location of the interaction, but David Tolby's lab invented this wonderful technique. There's an RNA ligation step, so you clone the junctions between a microRNA and an mRNA, for example. You can sequence across those as well. So you can talk about single reads and hybrid reads. I'll talk about both. So what we found in both cell types, with both approaches, is that W1 interacts mainly with protein coding genes, with mRNAs, but it also interacts with non-coding RNAs, including microRNAs. And the FLASH really revealed an interaction, this cross-linking revealed an interaction with microRNAs, and I'll come back to the relevance of that for tumours in a minute. This is very relevant for women's tumours, we think. Its interaction with microRNAs. So what about interaction with messenger RNA? Well, where it interacts is all at the 3' untranslated region. And the types of RNAs we have it interacting with in kidney cells are all things we think are logical: wound healing, epithelial differentiation, Wnt signaling, etc. The major categories that we get are the RNAs it pulls down are very logical for the biology it does. Now, when we look at hybrid molecules, we got a shock. The hybrid molecules we got, none of them were between RNAs hardly. They were foldbacks within the same RNA. So W1 is binding, and actually it promotes secondary structure from the 3' untranslated region over into the body of the RNA, or within the 3'. So it stabilizes these stem loop structures. And so the hypothesis is by doing that, it protects them from degradation. So W1 binding these, protecting them from degradation. So the first thing is you all talked about CHIP, and only a few things are regulated that the protein binds to in the chromosome. It's the same with RNA. when we delete WT1 in cell lines or knock it down, we get a few hundred RNAs changing. Some of them are down-regulated, and some of them are up-regulated. The problem is, if something's a transcription factor, how do you know it's not transcription regulating the levels of these RNAs, WT1's transcription function, or its interaction with the RNA? Well, interestingly, most of these RNAs are not transcriptional targets. Only 14%, 30 to 40% of these RNAs are those that bind to WT1, the ones that change in level when we delete it. And remarkably, all the RNAs that have foldbacks when you delete WT1, they all go down in expression. They don't go up. We have RNAs that go up that are transcriptional targets, but when we delete WT1, the RNAs that have the foldbacks go down in level. And to prove that these are what are some of the RNAs we get? The RNAs include R-spondins involved in Wnt signaling, insulin-like factor growth, factor binding proteins, polyphyllin, which is a crucial podocyte protein. These levels are all going down when we delete it in cell lines. But we're never happy with cell lines, so here we've taken real kidneys, kidney mesenchyme, wild type versus deleted, which we can fact sort, I won't go into that. And all these are also down-regulated. When we delete WT1 conditionally, these same RNAs go down. This is a log scale. They go down quite a lot when we delete WT1. These RNAs that bind to it. And to show it's post-transcriptional, this just shows a couple. What you can do is inhibit transcription, measure RNA stability, and the half-lives of these RNAs go down dramatically when we delete WT1. So WT1 is binding to RNAs, protecting them from degradation. And there's a lot more RNAs it's doing this to than it's probably transcribing. Some of the RNAs are both transcriptional and RNA targets. And so that leads us with WT1 regulates RNA stability through promoting 3' foldovers. And what we want to understand, of course, what's the physiological relevance, what do the two different isoforms do, and what else is it doing, what's the structural basis of all this, and what else is it doing downstream of transcription? Is it regulating translation, etc.? We're looking at that at the moment. But now, finally, I'll have a few more minutes. This is unpublished, the next thing. MicroRNAs, as you probably know, are made from pre-microRNAs in the nucleus. They are processed to pre-microRNAs, which in the cytoplasm are processed again to mature microRNAs. So what we found is that WT1 binds to a number of microRNAs, specific ones, and it binds again to the stem structure in pre-microRNA. So is that interesting? Is there any relevance at all? Well, the reason we got interested in that is last year, or last 2 or 3 years, new genes have been identified that are mutated in Wilms tumor. There were only a few up to a few years ago, but exome sequencing in Wilms tumors has identified these genes. And you'll recognize some of these. Drosha and Dyson, both involved in microRNA processing. DGCR8, again in microRNA function. DIS3L2, involved in microRNA function. So there are several of these Wilms tumor genes which were mutated, involved in microRNA processing. That's not true in many other situations. This is very unusual for Wilms tumor. Now, so we got interested in a specific one of these microRNAs, which is LET7. So LET7 microRNA is important for differentiation in a number of tissues, and it's a tumor suppressor. LET7 causes the degradation of LIN28, which is a pluripotency factor and can be an oncogene. And a paper I refereed and wrote an editorial of for Genes Development, a very nice paper, showed that if you overexpress LIN28 in Wilms tumor expressing cells during kidney development, you get huge Wilms tumors. But then if you express LET7 in the same mice, you don't get tumors. So LET7, LIN28 is an oncogene for Wilms tumor, and it's often expressed in high levels of Wilms tumor, and LET7 counteracts that. So WT1, actually, the best candidates for binding in microRNAs we have are LET7. And if in ES cells we delete WT1, we increase the levels of the pre-microRNA and decrease the levels of the mature microRNA. It binds to the precursor, you delete W1, take it away, you increase the pre. This is the normal level, and it's gone up sort of fourfold the pre-microRNA, and you've had a big reduction in the mature microRNA, LET7. And the same is true in kidney cell line. This is just a knockdown. We get an increase when we knock WT1 down in this kidney cell line. The levels of the pre-microRNA go up dramatically, LET7C in this case, and the levels of the mature go down. That suggests that binding WT1, we're testing this mechanistically at the moment, biochemically, it binds to LET7, helps promote LET7 processing. It's a post-transcriptional event. Has this relevance for any biology or... if we do a gain-of-function experiment, here in knockout ES cells, we induce with the doxycycline system WT1, we increase the level of mature LET7 RNA. That really supports the idea that it's regulating physiologically LET7. And that seems to be true in real tissue. If we look in real tissue, kidney tissue, where we deleted WT1, pre-LET7 goes up, mature LET7 goes down. But most interestingly is tumours. There are Wilms tumours which have either mutations in DICER or DROSHA, and you get LET7 levels going down. But if they're mutated in WT1, and not, these are human tumours, and not DICER or DROSHA, we also get LET7 going down. If the tumours don't have mutation in WT1, you don't have LET7, or the microRNA protein, you don't have LET7 going down. Several other microRNAs don't change. It's their specificity. This other one sort of does, mirror 181. So actually, both in kidney development and in Wilms tumours, WT1 seems to be important for the regulation of LET7 processing, and LET7 is a tumour suppressor for Wilms tumours. So this is an unpublished story that's getting close to completion. We need a bit more biochemistry yet. So my summary is, when people say, oh God, how can anything be so complicated as this? But this is an unusual protein, I think. So this WT1 can be involved in stem progenitor cell production, maintenance, or differentiation, depending on the context. It can do MET or EMT, depending on the context. It can be a transcriptional activator or repressor of the same genes. It can regulate tissue homeostasis and is reactivated after tissue damage and actually involved in tissue repair as well. It's not only a transcription factor, it binds mRNAs, regulates mRNA stability, and we predict it's going to be important splicing and translation. We're looking at that at the moment. It's not just a tumour suppressor, it's a fibrosis suppressor. And the last thing is it promotes microRNA processing, and that's potentially important in nephrogenesis and Wilms tumour. And I just, here's some of my colleagues. This is Yo Ying, who worked on the fat work, who's now an independent group leader across town in Edinburgh. She stretched this slide to make everybody look thinner. And she worked on fat. She didn't have to stretch it particularly, but she did. All the RNA work's been done by Selby here. It's fantastic. She's still in my lab and hoping to go back to India, just interviewed for some jobs. This is Abdul, who's now in a wheelchair. And he's finding life very hard. He's a great scientist. I just hope he can get back to it. And that's Peter Hohenstein, who's done a lot of work with me on, and he's now left in Leiden. He's now got a group lead position in Leiden. So just thank the people who've worked with us, lots of them. In Nice, Andreas Schlegel helped us do all the lineage tracing. Ramon is a real scholar in Malaga who's helped us understand development. And Jamie Davis helped us with the kidney. David Tollery helped us with the RNA interaction screens. And we're very lucky in the Institute. People help us with facts, Animal House staff, bioinformatics. We have these people to help us. They're so important. Oh, hopefully that was all right. We're all over the place. Thank you very much.